Okay. Good evening, everybody. My name is Britt Ranson Olson. I'm a professor of biology here at Lake Superior State University. And I wanted to just welcome everyone to our campus and this evening's Your Health Lecture series. This is a series that's brought to us yearly by Michigan State University College of Human Medicine, and LSSU being a community affiliate. We're proud to bring these excellent speakers. They're experts, in their area of medical research, other aspects of healthcare. And we're proud, to bring this knowledge to our campus, to the students, the faculty, but also the community. And we hope you, enjoy this evening. I want to introduce now Mark Breavy. He's the director of Community and Government Relations, again at MSU College of Human Medicine.
Thank you so much, and thank you all for coming tonight. We do have a virtual audience as well, so thank you for those that are joining us online. And I just want to say thank you to Dr. Olson for being such a great host once again. Earlier this evening, we had an opportunity for Dr. Counts to meet with some pre-med, pre-health students here at Lake Superior State University. Yesterday, we were in Escanaba at Bay College, and we've definitely have enjoyed our visit so far. So always great to be back up in the Upper Peninsula. And like Dr. Olson said, this is a long-standing relationship that we've had with Lake Superior State University, between Michigan State University College of Human Medicine and Lake State. And we look forward to coming up every year, hoping to do it again next academic year. So with that, I'm going to introduce Dr. Counts. Dr. Scott Counts received his undergraduate degree from Davidson College, concentrating in history and English. After working for several years as a chemist at the U.S. Centers of Disease Control, he went on to earn his PhD in neuroscience from Emory University, studying mechanisms of familial Alzheimer's disease. He then joined a lab at Rush University Medical Center to study cholinergic mechanisms of Alzheimer's disease and its prodromal stage mild cognitive impairment as part of Rush's NIA-funded training program in age-related neurodegenerative disorders. Following his postdoctoral training, Dr. Counts was appointed as an assistant professor of neurological sciences at Rush. And in 2013, he joined us here at Michigan State University College of Human Medicine as an associate professor of translational neuroscience. That's his primary and family medicine at the Grand Rapids campus. His current research focuses on molecular mechanisms of neuronal projection system vulnerability and mild cognitive impairment and Alzheimer's disease, as well as vascular contributions to dementia. With that, can you help me welcome Dr. Counts? Go on here.
Okay, can you guys hear me okay? All right. Yeah, thanks, Mark. I appreciate the warm introduction, and it's great to be here after 3 years. Last time we were here, we were all here just a few months away from our lives being turned completely upside down by COVID. So it's good to be back to normal a little bit. And it was great. We got here early and I was able to stroll around the campus, and it's a really beautiful campus, and take pictures of Canada, stuff like that. Want to talk a little bit to you about Alzheimer's disease this evening, and let's see if we can. Okay, and then can you guys see the pointer? Okay, everybody. Okay, good. All right, so just our basic agenda here. We're going to talk a little bit about some brain basics, just to get everybody on, kind of on the same page. Some basics about just dementia as sort of an umbrella term. Then we'll move into Alzheimer's disease in particular, its characteristics and socioeconomic impact. And then we'll talk about the latest in diagnosing the disease, from everything from imaging people, in person to blood tests. And then we'll talk about the latest in therapies, what's in the pipeline, what to think about this drug that was approved last year that was real controversial. And then many of you may have seen in the news that there's a new drug that just showed some promise, and I'll tell you what I know about that. And then we'll talk about risk factors, because right now, of course, we don't really have a treatment for Alzheimer's disease. So, what are some things in the interim that we can do to reduce our risk? Okay. And so let's start with some brain basics. And so here's your, the human brain, and lots of different compartments of the brain that do take care of different functions for us. So we have down here the brain stem, the little stem here that connects to the spinal cord. This area of the brain from a cognitive standpoint is involved in attention and vigilance. And actually there are nuclei in the brainstem that degenerate in Alzheimer's disease. And that probably impacts our attentional abilities in the disease. And then this area is also important for autonomic function. So, controlling your breathing and your heart rate, things that, you just do automatically. So you don't have to think about the fact that you're doing them. And then here's our cerebellum underneath in the back here. This is our center for balance and coordination. And so, when we're watching our favorite sports teams, they're obviously using their cerebellums over time, but, we need them just to walk down the hall, right? So obviously a very important part of our brain. And then the large portion here is the cerebrum, and it's divided into 4 primary lobes. We have the occipital lobe in the back. And that's actually, even though our eyes are, of course, in the front, the occipital lobe is in the back and controls our vision. It's basically in charge of taking the sensory input that we're getting through our eyeballs and then telling us what it is that we're seeing, right? And then here, down here, sort of behind the ears, is the temporal lobe, right? And so that's our area for memory and language. And this is an area that does degenerate very early in the disease, in Alzheimer's disease. And then up here in the front, sort of, behind your forehead is the frontal lobe. This area is important for what we call executive function, like planning and making decisions and solving problems. It's also an area for our personality and our general behavior. And then lastly, sort of back here and sort of more towards the crown of your head is the parietal lobe. And this is an area that sort of integrates all of our sensory input that we're receiving, makes sense of that, and then tells us sort of how to react to that input, and sort of creates how we react to what we're doing in our everyday lives. And so now if we take the brain, so now the students, of course, have probably been working with microscopes quite a bit, and maybe the rest of you remember this from school or what have you, but so if we take a human brain and we cut it super thin and put it on a microscope slide and use a special stain and look at it under the microscope, we can see what's sort of the fundamental cell of the brain, and that's called a neuron. Okay, and so you see it's a very beautiful structure, a nice little cell body, and all these sort of branches coming off of it, right? And so in fact, we can almost envision it as a neuron forest in the brain. And so, you see, of course, all these branches here out, and these branches all communicate with each other, okay? And they sort of talk to each other using these special connections called synapses. And synapse is just a Greek word that means come together. And so these synapses, so one neuron can talk to the other by secreting a chemical messenger, and that neuron receives the message, and then so on and so on. It's like a game of telephone. And that's sort of how the brain communicates with each other. And this is a 3D reconstruction of all the different connections that are going on in the brain. Okay, and so the complexity of us as human beings is basically based on all this structure. So we have 100 billion neurons with over 1,000 connections each of these synapses each. Okay, and so that's 100 trillion connections or synapses. Okay. And so that's what makes us so complex as human beings. It was, we were just talking about this yesterday. I was actually sitting in the front of the plane on the way into Delta County Airport. And, I was just sitting there staring at the cockpit going, how on earth did they figure out how to do that and then actually put a plane up? And that was, 100 years ago. Now, 50 years ago, we're putting people on the moon. Now look at all the stuff we're doing. It's just amazing. And so that's owing in part to the structural complexity of our brains, okay? And so congratulations, we're now all neuroscientists. My work here is done. And so let's talk about dementia basics. So dementia is thought of as an umbrella term, okay? And what it's referring to is really anything, what dementia is a decline in memory and thinking skills that disrupts our daily lives, right? And that's caused primarily by damage to these brain connections, right? That control knowledge and memory and attention, judgment, problem solving. and language. And just to sort of give you an idea of sort of the problem that dementia is going to become for us as a global society, it's predicted that by 2050 in the Americas, we'll see about a 250% increase in the number of people with dementia, similar increases in Southeast Asia, much larger increases in Africa, and actually I would include the Middle East in this. And a lot of that is really people with, as there's an increased educational attainment and increased modernization, there's also increased exposure to a lot of the risk factors that are associated with dementia. And then Europe is going to see a smaller increase, and that's basically based on just their population not growing as fast as some of the other continents. And so these are the faces of dementia, right? And so we see a lot of these faces that we know. Some of us older in the audience recognize some that maybe the younger ones don't, and vice versa. But dementia is really a great equalizer, right? It affects the rich and famous and people that who have policies that affect our lives and all of this, and that's great. It gives it a public face and creates awareness. But the people that we're really more concerned about on from a day-to-day basis are our own families, right, and our own friends and their families, and so this picture in the middle is actually my grandmother's engagement photo, and she had a vascular type of dementia back in the about 50 or so years ago. So again, this is something that affects all of us, and that's why it's really a concern for us in our lives today. And so let's move into Alzheimer's disease and give you a brief overview of that. And so this is a picture of Dr. Alzheimer and a picture of his first patient, August Dieter. Actually, not his first patient, but this is the first patient that he described in his scientific papers and presented at conferences. He studied her while she was alive, and then when she died, he studied her brain and discovered what is basically the pathology that we now refer to as Alzheimer's disease, as characterizing Alzheimer's disease. Okay, and Dr. Alzheimer kept notes on Ms. Dieter, and one of the things that he wrote was, when she has to write her name, she's unable to do so, and keeps repeating, I've lost myself. And I think those 4 words actually encapsulates what the disease means for the patient, right? You're disconnected from your past. You have only a fragmentary, reaction to the present. You're watching as all these memories that you've built up in your life are sort of slipping away, and you just lose your identity, right? And so that's, I think, the key to this. You lose yourself, and of course, this is, it's devastating for the patient. And of course, it also, this impacts our loved ones and people are caring for us as well. And what we know about Alzheimer's disease now is that it's the most common cause of dementia in the elderly. Okay, so again, we've got that umbrella term of dementia. Alzheimer's disease is the #1 cause for that dementia. It's about 60% of the cases here or there. And what distinguishes Alzheimer's disease from other types of dementia, and this is for maybe some of the pre-med students too, as far as clinical differentiation, is that it begins with the progressive memory loss. It's sort of a pure memory thing, right? And attentional problems. And then you move into issues like impaired language and activities of daily living, executive function, and then ultimately into sometimes some behavioral changes. Then, of course, we know the disease right now, once it starts, it's an inexorable decline. And so it's irreversible. And that's obviously the biggest challenge for us as researchers and as a society dealing with this disease. And I put these other 2 pictures up because just as a sort of a historical aside, so Dr. Alzheimer wasn't the only person studying patients with dementia, and he wasn't the only person looking at their brains under the microscope. And so about the same time that he was doing this, other people were, and 2 of the more prominent ones were Dr. Oscar Fischer, who was at the, so Dr. Alzheimer was in Munich. Actually, August Dieter was at a place in Frankfurt where he was visiting her. Dr. Fischer was in Czechoslovakia. And Solomon Fuller, who's really has gone sort of unrecognized, was an African-American. His parents were from Liberia, and he was working at an asylum in Massachusetts because that's where dementia patients went, 100+ years ago. And so he actually presented a paper on the disease at a conference at Clark University that was attended by Sigmund Freud and Carl Jung and all these famous people. But what happened was Dr. Alzheimer's mentor wrote a book, textbook, went to a major European conference and just started calling it Alzheimer's disease. And so that's how we know of it today. So I just thought that was a little interesting. And it's just important to remember that, you know, a lot of people were looking at this at about the same time. And so when we look at sort of the global brain changes in Alzheimer's disease, it's what the disease really is a degeneration and a loss of those neurons and those connections, those synapses, right? And the loss of these neurons and connections occurs preferentially in these brain areas that control memory and attention, those executive functions like planning and problem solving and language, right? And so what that results in is atrophy or shrinkage of the brain. And so, you know, here's a healthy brain here on the top left, and you can see this in a cross section. And then here's the Alzheimer's brain, and you can see all the narrowing, the gaps in between the folds as those neurons start to die off and the tissue starts to shrink. And then you can see it in the cross section. It's a very obvious change. These big sort of tunnels here are called the ventricles. We'll talk about that a little bit more, but that's where the cerebral spinal fluid goes. So as the brain shrinks, those ventricles become larger, and this sort of these white areas, that's the white matter. So that's where all these connections are going back and forth between the brain. And as you see these white matter areas shrinking, that means those connections again are being lost, right? And so one area I want to show you in particular, though, is the hippocampus. So the hippocampus is the main area for memory storage in the brain, okay? And so if you can appreciate, the reason it's called hippocampus is because the anatomist that discovered it thought it looked like a seahorse, and hippocampus is the Greek word for seahorse, which is kind of cool. And so here's the hippocampus in the human brain, and you can appreciate just how decimated it is in the Alzheimer's brain, in the advanced Alzheimer's brain. And then this sort of cartoon up here again shows you that the degeneration is really more selective for these areas of like memory and language and places like that. Okay. And now, you know, when we pull our microscopes back out and we take thin slices from the brains of Alzheimer's patients, and we use, again, special stains, what we start to see are the two major pathologies of Alzheimer's disease. And these are, and you may have heard of this in your reading or whatever, these are the plaques and the tangles, right? And so with respect to the plaques, they're actually called amyloid plaques, okay? And these plaques build up between the neurons. So if you remember the picture of the forest, they're kind of out there where the branches are, and not inside of the neurons themselves. And this is a picture that Dr. Alzheimer himself actually drew of these when he was looking under the microscope at August Dieter's brain. And he called them thickened miliary foci, which I don't really don't understand what that means. But anyway, that's what they were calling it back then. Now that we have all of our modern techniques, of course, we are able to really understand what these plaques are. And now we know that the plaques, and these are plaques out in the frontal lobe, these are made of aggregates of a protein called beta amyloid, or A-beta. And I've got these underlined because these terms are going to come back in some future slides, but they both basically refer to this protein called amyloid, okay? And these are just aggregated like little gumballs out in the brain. And what you can see is, so this dark stain is where the amyloid is. And you also see dark stain in these little tubular structures. Well, those are blood vessels. And so you get amyloid building up in the blood vessels too. And so that, starts to interrupt blood flow in the brain. And I just wanted to show this. So this is Oscar Fischer's drawings of plaques that he was producing at about the same time as Dr. Alzheimer. And so maybe not quite as an accomplished artist, but he was definitely drawing, similar structures, right? And then our other major pathology are these tangles, but more specifically, they're called neurofibrillary tangles, okay? And these build up actually inside of the neurons, okay? And so again, these are drawings from Dr. Alzheimer, these sort of wispy structures inside of the neurons. He called them peculiar fibrillary changes of the nerve cells. Again, with our modern techniques, we now know that these tangles, and this is shown in the hippocampus, that memory storage center. These are made of aggregates of a protein called tau, so the Greek letter T, tau. And again, I underline that because, well, that'll come back to us too. But when you tau, and so it's aggregates of the tau that make these tangles. And so all of these neuron structures here that are brown and black, those are filled with tangles. So these are sick and dying neurons in the hippocampus. So this is not what you want to see, basically, right? And again, just to show you, just for historical perspective, these are actually some drawings from Solomon Fuller that were made at about the same time that Dr. Alzheimer was making his drawings. So again, that these tangles were recognized by many at the time. And so, the bright side, and again, this was just a very brief overview, obviously, but so the bright side of this, of course, is that we do understand many aspects of Alzheimer's brain changes, right? We know we get these amyloid plaques, and if you can sort of see this scale, so green is less severe and red is more severe. And so you can sort of see how, over time, these plaques spread throughout the brain and get really severe, like in these areas up here. And then the tangles, they do the same. They sort of start at that sort of hippocampus type area. And then they, of course, then start to spread and get more severe over time. And then, this leads to our neuron loss and disconnection of those synapses. And then we start to see the shrinkage and atrophy of the brain. And then we get these symptoms, right? The memory loss and the executive functions and all these other issues. And so that's the bright side. The not so bright side is that we still just don't know enough to develop an effective therapy to slow down the symptoms, right? And so that's the big challenge, right? And so one of the main reasons this is a challenge is because of the current socioeconomic impact of Alzheimer's disease. And so this is facts and figures. There's actually an entire PDF of this, but they also have sort of this broadsheet that you can print out or download, and you can download it this is from the Alzheimer's Association, so it's just alz.org. And just some of the numbers here I want to highlight is that, there are currently more than 6 million Americans living with Alzheimer's disease. Over the past 20 years, there's been about 145% increase of people who've died from Alzheimer's disease, whereas folks with heart disease there have been decreases in deaths due to heart disease. This is a very important figure. This is talking about caregivers, and these are really sort of the second patient of the disease. So currently there are over 11 million Americans providing more than 16 billion hours of unpaid care, valued at $272 billion. So you can see how what an economic impact that has on people that are, say, coming out of the workforce to take care of someone, right? And then a couple more. Just this one over here, and so in 2022, Alzheimer's and other dementias are going to cost the US alone $321 billion. That's projected to increase to $1 trillion by 2050. And one of the reasons these numbers are particularly important is the impact on Medicare. And it's maybe a little bit hard to see, and this is actually from 2017, but the numbers are pretty similar today. If they've maybe just gone up some, right? But what you can appreciate is these are payouts to beneficiaries with Alzheimer's and other dementias here on the left column. And on the right column, we have beneficiaries without Alzheimer's or other dementias, right? And so you can see three times as much money is going to the Alzheimer's beneficiaries for inpatient healthcare, more for Medicare medical providers, way more for skilled nursing facilities and nursing home expenditures and so on. And so, when you think about Medicare as a limited resource, right, the larger that pie is going to care for Alzheimer's and dementia patients, the less of the pie there is for people suffering from other diseases, right? And so that's we need to, be able to slow down the disease so we can start to, narrow that slice of pie a little bit, so we can start to have the flexibility to take care of everyone. And so let's go into diagnostics now. And so, again, the key for diagnostics now is finding people in the early AD stages, because that's going to be the key to an effective therapy, right? Think about cancer, right? You want to find people in stage 1, not stage 4. It's the same idea with any disease, really. Finding it earlier, you have a much better chance of a treatment being successful, right? And so when we think about diagnosing Alzheimer's disease, what we're thinking about now is trying to find what we're calling these preclinical stages, okay? So before people actually have symptoms. And that's difficult to do because they don't have symptoms. How do you know they have the disease, right? And so we'll start with this basic graph here. And so this is on over here, this is sort of high functionality and low functionality down here. And then we have our lifespan, and most of us hope we sort of follow along this yellow line here and wind up with normal age-related memory loss, right? And unacceptable to many of us will veer off of this curve, become, have Alzheimer's disease or some other type of dementia, and ultimately wind up with a total loss of independence and loss of function. And so where we're targeting now are these people that are in the clinic, at least, are people that are just starting to show these memory problems, and they're referred to as having mild cognitive impairment. This word amnestic here, amnestic is just the adjective for amnesia, right? So you think of amnesia, losing your memory. And so that's what's happening. These folks, they're having these memory difficulties, but they're still able to function okay. Their brain is actually compensating. But again, your treatment is probably much better, this part of the line, the line right here, than down here, okay? Now, one of the problems with mild cognitive impairment, though, is that what we're finding in postmortem brain tissue is that people who come to autopsy with a diagnosis of mild cognitive impairment often show up to 25 to 30% loss of neurons in these cognitive brain regions like the hippocampus, right? And so, which is, if you think about it, the brain's an amazing thing. It can handle all this cell loss, and you still only have very slight issues, right? But it creates a problem for trying to diagnose people because, again, by the time they're showing symptoms, even the slightest symptoms, there's already a lot of things going on there. And so what we're trying to target now are these so-called preclinical stages, right? So it's now believed that Alzheimer's may begin decades before symptoms, right? And so identifying people in the preclinical stages, and yes, even MCI, I mean, because, you've got, you stand a much better shot here than down here, right? That is going to be key for enrolling people into clinical trials, and that therefore these therapies have a much better chance for success, okay? And so, again, like I was saying before, if we can find you earlier, treat you earlier, the chances of success are going to be better. Okay, and one of the ways they're doing this is with imaging. And I know this is a busy slide, but we can talk through it, no problem. And so everybody's heard of PET scans, right? Yeah, and so positron emission tomography, kind of the official name for it, but the PET scans involve putting a sort of what they call a tracer into your body that goes and identifies some problem in the brain or the body, right? If you're looking for tumors, for instance, and then you'll see like a hot spot on the scan, identifying where these problems might be, right? And so now they have special PET scan tracers that can go into the brain and bind aggregates of the amyloid, the A beta, and so that's finding the plaques, okay, in the brain. And then they also have these tracers that can go in and find the aggregates of the tau. And so that's finding the tangles. So now we have these, we can use PET scans to find plaques and tangles in living subjects, which is really cool. And so let's take a look at these 4 subjects here. So we're looking at their amyloid, we're looking at their plaques and their tangles. And so here's a person in the early 70s. And again, when we look at these images, you think of the cool colors as very low levels of the pathology, and these warm colors are very high levels of pathology. Okay, and so this person on the top here, cognitively normal, they just have very little signal for anything, and so that's probably a lot of this blue maybe actually just be background. Okay, but then compare that to this person in their 70s that has dementia. I mean, now their brains are just lit up with these plaques and lit up with these tangles. Okay. Now let's look at 2 normal people in their 80s here in the middle. So look at the person on the top there. They look more like the person on the very top, the normal person, right? And then here's a person with no cognitive problems, and yet look at all the plaques and tangles in their brain. So this is a person we would define as having preclinical Alzheimer's disease. They're still normal, but they obviously have a lot of pathology. So that's the kind of person that's like, okay, let's get that person into a trial. Likewise, you can use what's called the FDG PET, and that detects the glucose uptake. And so this is the typical PET scan you would get, for instance, to try and detect a tumor. And so what's going to happen is where you have lots of cell activity, like in a tumor, there's going to be a lot of glucose uptake because those cells need that energy, right? The opposite's going to happen in the brain during Alzheimer's disease. As the neurons die off, a particular area of the brain may have less of the glucose uptake. And so it actually you see a reduction in the signal. So instead of a hot spot, you actually see a cool spot. And so these are two people that both have no cognitive issues, right? A and B. And at first glance, these brains kind of look the same, but if you look there at the top, right? So you can sort of see where now we're getting these sort of cooler signals in this person on the right, subject B, compared to subject A, and sort of along the sides here, see? And so that person that could be another marker for someone who has preclinical Alzheimer's disease. Those areas of their brain are already starting to sort of die off, right? And then we can use, just the good old MRI, where we can actually see structurally what the brain looks like in a living person. And again, you can appreciate the difference between this cognitively normal person and here's this person with that amnestic, the memory-related mild cognitive impairment. You can start to see there is evidence for some little bit of shrinkage there, right? Nowhere near as much as the person with Alzheimer's disease, though. So again, this is a good person to try and target for therapy. And generally what we see is that it's the amyloid PET over here. Those are sort of the first signals that we see. So in a lot of the clinical trials, as we'll see, a lot of the selection that they use to enroll people is that they do have what they call a positive amyloid PET scan. So when they put these people in the PET scanner, they do see the plaques. And that sort of tells the clinicians that yes, this person is probably has evidence for Alzheimer's disease or eventual Alzheimer's disease. And so another way that we can diagnose Alzheimer's preclinically is looking at changes in the amyloid and the tau in our cerebrospinal fluid. Okay, and so the cerebrospinal fluid, it's runs up and down your spinal cord, and then it goes up into your brain, and it goes through those ventricles, those little tunnels that I was showing you earlier, right? And what it does, it just bathes the brain and exchanges nutrients. It removes toxins. It's almost like the motor oil, for the brain, just to keep things running smoothly. And what people and the way to isolate this CSF from a living subject is through a spinal tap, a lumbar puncture, right? And so what the researchers have found is that people who eventually convert to Alzheimer's disease, okay, they'll display, years prior to their clinical symptoms, they'll display a rapid annual decrease in amyloid in their CSF. And the reason they think it's decreasing is because it's depositing into the brain. And so it's just sticking there and it won't get into the CSF. So it gets less and less of it as the plaques accumulate. And then what you see then are also rapid annual increases in tau in the CSF. And the theory here is that because remember the tau, those tangles are inside of the neurons, right? And so when those neurons die, they break apart, the tau sort of spills out and it gets into the CSF. And then the more that happens, the more tau they find in the CSF. And so this is these are actually really viable markers to use. The only problem is, most people don't want to get a spinal tap every year, right? I mean, I certainly don't. And so now sort of the grail, if you will, for the field is a blood test, right? And so, for some of the reasons are pretty obvious. It'd be much cheaper, much easier to administer than PET or MRI scans. Not everywhere has PET or MRI scans, and the interest, the thing about the PET scans is that you actually need special facilities close by who are actually going to make those amyloid and tau tracers so that's really confined to, like University of Michigan for us and some of the major medical centers like in Chicago and places like that, University of Wisconsin too, for that matter. And then of course, it's also going to be much less invasive than collecting CSF, just a little blood draw, whatever. And then it's also much more practical for community-based medicine and for just smaller communities in general, right? So you can have a screening if the markers in the blood seem like a little off, like they look like, oh well, there could be something going on here. Then you can get referred to a specialist and start exploring that possibility further, right? And again, the whole point is especially with screening, is to find people who might be in the early stages. So when we treat them, they have a better shot of having a successful outcome. And right now, the lead candidates, at least from a preclinical AD standpoint, is we see sort of a lower, it's sort of a ratio of amyloid in the plasma. And then we see a higher, what's called p-tau, and that's just a special type of tau that's involved in the formation of the tangles. And then we also see higher what's called a neurofilament light. And that's just a marker for neuronal death. So again, as the neurons die, this neurofilament light sort of spills out into the, gets into the bloodstream, and then you just see more of it as more neurons die. And so these two figures here I want to show you, the point of these figures is not to, do a deep dive and really try and, understand exactly what's being shown here and how to interpret it, but just gives you examples of what we see as researchers and what's some of the research that's coming out just this year, okay? And one thing so what they did here is if you look around, this is amyloid PET down at the bottom here, right on the left here. And so these people in blue were negative. So they did the scan, no evidence of amyloid. The people in the red here, they did have positive PET scans, right, for the amyloid. Now in their blood, the blood showed in the positive PET scan people, this special ratio of the amyloid, okay? And so what that's telling us is that, just looking at this amyloid in the blood may be just as reliable as looking at doing a PET scan, right? And so that's great, because now we can get just screen through a blood sample and not have to worry about, finding a medical center that can do the scanning. And then over here, now we're looking at what's called, the p-tau, the special form of tau. And what these are along the bottom here, these are just, so this is normal controls, people with mild cognitive impairment, people with Alzheimer's disease. And these are folks that have different neurodegenerative diseases that cause dementia. So there's corticobasal syndrome and frontotemporal dementia, and a disease that's known as primary progressive aphasia. But what's important here is when we measure this sort of tau, again, it's another ratio, we measure this tau in the plasma, what we can find is that, it's very selective for Alzheimer's disease. And it's actually increasing the worse a person's condition gets, right? And so it not only does it tell us, maybe preclinically, if if it looks like you have Alzheimer's disease developing, but it can also, like as just you pre-med students, it can help you differentiate whether a person is going to have dementia based on Alzheimer's disease or maybe from another source. So that's kind of cool too. Okay. Okay, and so that's diagnosis. And now what's the latest in treatment? So the goal obviously is, as we like to say, is to make Alzheimer's disease a distant memory, right? So the play on words there, but and in all likelihood, the more realistic goal is to have a disease that sort of what we call modifies the progression of the disease and just slows it down, right? So if you can just slow it down 5 years, 10 years, that would be an enormous impact on everyone. And so let's start with this drug called Aduhelm that was it was in the news a lot the last couple years. And it was actually FDA approved. It was the first FDA approved drug in like 20 years for the treatment of Alzheimer's disease. And so that's what made it a big deal, but it was very controversial. And so what Aduhelm is and it's actually, its trade name is, well, that's the trade name. What it's really, its scientific name is aducanumab. So, these drug companies try and come up with the most complicated names possible for these drugs, but it's aducanumab. And what these drugs are antibodies to amyloid. And so what they do is, again, it's infused into your bloodstream, the antibody goes up into your brain, and the antibody finds the plaques, and it starts to destroy the plaques. And so the idea is if you get rid of the plaques, you can make their symptoms better and actually maybe even slow down the progression of the disease. Okay, and so what happened here with this aducanumab, or the Aduhelm, is that they had 2 phase 3 trials, and they were identical. So and phase 3 is when you have like thousands of people in the trial, and it usually goes on for 18 months, 2 years. They want to make sure, they want to make sure it continues to be relatively safe. And most importantly, in the case of Alzheimer's drugs, they want to see that it improves the clinical symptoms or slows cognitive deterioration in the people that are getting the drug compared to the people that are getting the placebo, or the sugar pill, as we always like to say. And again, the goal here isn't to do a deep dive on these graphs and everything, but again, this is just the way the data is presented. And what it's showing is that they had these 2 trials. One was called the Emerge trial, one was called the Engage trial. Okay, and what the Emerge trial showed was that and again, it's just the difference between this dot and that dot, okay? Is that what that represents is a 20% less decline in global cognition with the highest dose compared to the placebo after 2 years. So had baseline, one group gets the high dose, one group gets basically nothing. And then 2 years later, the people with the highest dose have shown a 20% less decline in their cognitive skills. So that, it's obviously very modest, but at least it's something, right? But the other trial had no effect at all. So it didn't even work. And so now you have this drug that worked in one trial, but didn't work in another, okay? The other issue here was when they looked at side effects. And so one of the side effects that they see, and this is a little esoteric, but it's called ARIA. And it refers to amyloid-related imaging abnormality. That's sort of the acronym. And it's just what they're seeing. They're just seeing weird signals on the brain scans, right? When they do these amyloid PET scans. And what they've actually come to understand is that there are two types. There's one called ARIA-E that actually they realize now means there's brain swelling in response to the drug. And then there's ARIA-H, which means the brain's actually bleeding, possibly in response to the drug. And again, this is the chart. As you can see, we're talking about over 1,000 people in the placebo and 1,000 people in getting the drug. And what they found were like 35% of the patients were showing brain swelling with the drug, and 35% of the patients were showing brain bleeding with the drug. Okay. And so this went to so that's not insignificant, correct? And so this went to the FDA advisory committee, and the advisory committee is made up of the top Alzheimer's researchers in the country, basically. Many of them were actually running these actual clinical trials using this drug. And then also, importantly, some of the top statisticians in the company, in the country, who can look at the company's data and make their own conclusions about what the data actually mean. Because it's human nature to take your data and present it in the best possible light, right? So that's why we have peer review. So we can call you out. So the questions that they asked, two questions. Does the eMERGE study viewed without regard to the ENGAGE study? So in other words, does the study at work, if you think about just about the study that worked and totally forget about the study that didn't work, does that give us strong evidence that the drug is effective? 1 said yes, 8 said no, 2 uncertain. So there are 11 people on the panel, like tiebreaker, right? The next question was, is it reasonable to consider the successful trial as sufficient evidence that the drug is effective for the treatment of the drug? That time, a resounding no. But the FDA, who was under a lot of pressure for this drug to succeed, because again, there's such a dire need for a drug that works, decided just to postpone and wanted to collect more and ask them to collect more data. And what they did find that was actually interesting and actually pretty impressive was they found that aducanumab also lowered tangle pathology. So now we're, instead of doing the amyloid PET scans, we're doing those tau PET scans, looking at the tangles. Okay, and so here's a person who got the placebo, they're a control, and here's a person that got the drug, okay? And now, again, remember, we got the hot spots mean there's more, these cool spots mean there's less. And so if you look at this patient, from baseline to follow-up, you can see these, you're sort of starting to get these hot spots getting hotter, right? The more red as on follow-up. So this tells us that this person's, and even up here, so this is telling us this person is actually continuing to accumulate these tangles in their brain. But the people that were getting the aducanumab, they were finding, so look how red that is, and then look how more like orange that is, and look how that little red spot goes down here. So they were actually, it looked like the tangles were actually being reduced by the drug, and so that's a good thing, right? And so the FDA granted this accelerated approval. Okay, and so it's obviously, like I said, a drug for a serious illness with an unmet need. Their rationale was that the fact that it can lower the plaques and tangles means it's reasonably likely to have a clinical benefit. But the caveat that they threw in is that they wanted to see more trials to verify that there's actually a clinical benefit and acceptable safety. And so, there's always the chance they could just take it off the market if the trials are negative. And so that created a little tension. So what happened were 3 of the advisory panelists actually just resigned. Here's some interesting comments that aren't necessarily positive. And then again, many of the medical centers, including those in Michigan, just decided not to offer the drug as a treatment option. If you'll recall, the initial cost was like $50,000 or something a year, and then they slashed it, down to $25,000 or whatever it was. But what happened was Medicare stepped in and said, and they sort of split hairs too. It's like, okay, we'll cover it, but only if you're for the people who are participants in these new clinical trials, right? So unless you're one of the couple thousand people that are in these clinical trials, few thousand people, it's $25,000 a year, right? And so it's become, now it's a drug that most people can't afford, and it's a major health equity issue, right? So that's where we are with the Aduhelm. Let's talk about some good news, okay? So what happened while all this craziness with Aduhelm is that the National Institute on Aging, the NIA, decided to launch this clinical trial consortium, okay? And now what they're doing is they're focusing on early preclinical stages of the disease. So now we're talking about prevention, not rescue, like not giving the drug to someone with Alzheimer's, but trying to give it earlier, like we've been talking about, right? And then what they're doing is they're going to fund, so again, instead of the drug companies funding it, your tax dollars at work, okay? And what they're doing is they're funding 5 to 7 5-year trials, and they're being funded based on peer review. So in other words, it's not just the drug company testing out their drug. This actually has to pass rigorous peer review before they decide which drugs to actually test, okay? And so the drug they decided to test first is this drug called BAN2401. And this is another antibody that it does the same thing as the aducanumab. It's just directed at a different type, but not type, but different structure of amyloid, if you will. And so there are definitely some differences from a sort of a scientific standpoint, if you will. And what they've done is they now have 1,400 subjects, and it's going to be a 4-year trial. And in one of the trials, they're going to just take normal subjects who have a little bit of a PET amyloid signal in their brains. They have some plaques in their brain, but they're still sort of what they call below threshold. So they don't really they don't really meet that threshold of what we call preclinical AD. And again, those thresholds are set by analyzing the brains of thousands of people. And what they want to see is if it can just like lower or prevent the accumulation of the plaques and tangles based on those PET signals, okay? The other trial, now they're taking normal subjects who do have the high amyloid PET signal, a lot of plaques in their brain. And then again, we're calling these the preclinical AD. And now we want to see, does it actually improve cognitive decline, similar to what they've been doing with these other trials, okay? And so that was just launched last year. We're humming along. And then late breaking news about BAN2401. And this is what we've been seeing in the news. The name you've been seeing is lecanemab, same drug as BAN2401. Okay, they're just, this was what it was called when it was being developed. This is now sort of its official name, if you will. And then if it ever makes it to market, there'll be some name like Aduhelm or whatever. So what this, and the name of the trial was the Capture AD trial, and it was an 18-month study, high dose of this, antibody that goes up into the brain, finds the plaques and chews them up. It is in 1,900 subjects, and they had early AD. And so by early AD, they had a positive amyloid, positive plaque signal, and a global cognitive score. So their cognition, their abilities, their cognitive abilities were similar, or were basically meant they were diagnosed with either mild cognitive impairment or just, really mild AD. Okay. What they found was that the drug slowed cognitive decline over the 18 months by 27%, and also reduced the plaques, which is what the other drug did. But now it's at 27%. One trial that was one big trial that was successful, not one that was successful and one that was unsuccessful. And then there was a reduced occurrence of the brain swelling and the brain bleeds, that ARIA that we were talking about, compared to the Aduhelm drug. Okay, and so again, this is the drug company reporting on this, getting out the PR, they're getting people excited, putting pressure on the scientific community, et cetera. But the results are actually going to be shown to the scientific community in about couple months at this Clinical Trials for Alzheimer's Disease conference in San Francisco. And then that's going to be when that's when the rubber hits the road with this. And we really see, once it comes under scrutiny and peer review, how well it's holding up. But a lot of people are excited about it because compared to this first drug, which ironically enough is coming from the same drug company, the same drug company partnership. But if we looked at percent slowing of cognitive decline, the eMERGE trial was, one was 20%, one was 0%, and then we had like 35% major side effects. This one is 27%, and only like around 12% side effects. So that's an improvement, and only time will tell if this makes it to FDA approval and things like that. But again, any news like this is obviously going to be some good news. Where are we with time? Okay, I better speed up a little bit. So what about tau? So I'm just going to tell you about one of these. And so, we've been talking about amyloid the whole time. What about tau, the parts of the tangles? And so this is just an interesting story, and then we'll move on. So this compound called LMTM, and what it does is it just prevents the tau from aggregating into those tangles inside of the neurons. Okay, and what they found was they were doing a study, and they found that the drug was actually failing, right? But one of the reasons that what they found was that the drug itself actually is sort of a blue dye. And so the people that were getting the drug, their urine was blue, and the people that got the placebo, there was fine, right? So basically you knew whether or not you had the drug, whether or not you had blue pee or not. And so that's a major problem because it affects how people behave in a clinical trial. And so what they did is for the control group, they just gave them a low dose. So now everybody's peeing blue, basically. And what they found, I don't think they expected it really, was that the low dose was actually what was creating an improvement in the subjects in the trial, not only for AD, but there was also a separate trial for frontotemporal dementia, which also involves some of the tangle pathology in some cases. And so now they have some new phase 3 large trials to look at that. And then there are just some antibody trials for tau that are also in the works. So again, going up into the brain, finding the tangles, and then trying to chew those up. And so what else is in the pipeline? Well, right now there are 31 agents that are in phase 3. So again, thousands of subjects. 68% of those are disease-modifying therapies. So therapies designed to slow down the disease. Of those, again, lots of amyloid. We've got our one tau, but then we're trying other things. We're trying to protect those synapses. You've heard of oxidative stress, so we're trying to target oxidative stress. The vasculature, trying to keep those blood vessels running smoothly, right? Looking at trying to keep the cells active and energetic, and even looking at inflammation, which is a major issue in Alzheimer's disease as well. We just didn't have any time to go really into that. And then in phase 2, where we have like hundreds of subjects, they have 82 agents that are in phase 2, and 87% of those are disease-modifying therapies. And again, still the amyloid. Now all these tau trials are coming online. A lot more trials looking at inflammation. Okay, and then again, protect a lot more. Trying to protect our synapses and some of these other potential contributors to Alzheimer's too. And then the other drugs are looking at trying to treat just the neuropsychiatric symptoms of people that already have the disease, or they're trying to just do like cognitive training type interventions just to kind of keep the brain more active and all that. So that's where we're at with treatments. So what about risk factors? How can we reduce our risk? Well, about 5 years ago, the Lancet Commission, which was a group of international experts, they reviewed all the data, and they found that there were 9 modifiable risk factors across the lifespan that, if you address them, would collectively reduce dementia risk by 30 to 35%. And it didn't even include diet, and we'll get to diet in a minute, but that's an interesting thing to note. So early in life, less education. In midlife, hypertension or high blood pressure, obesity or hearing loss. In later life, depression, diabetes, physical inactivity, smoking, and low social contact. And then last year, they sort of went back through and reviewed all the data again 5 years later, so roughly. these other 9 still held up, but they actually found 3 more, and that's traumatic brain injury and alcohol abuse in midlife, and then interestingly enough, air pollution in late life. So, and again, how these risk factors actually contribute to Alzheimer's disease, there's still a lot of, for many of these it's still not exactly clear, but that seems to be the case. And some, seem sort of straightforward, but, and one of the reasons this is important, risk factors, well, let's go back. So another risk issue, and again, these aren't modifiable risk factors, right? So these are more biological risk factors. And so, women and minorities are at greater risk for AD. Okay, so what this figure is showing us on the left, and this is the percentage risk for developing dementia later in life. As you see here, at age 45 and age 65, women are almost twice as likely as men to develop dementia or Alzheimer's disease. And then when we look at ethnicity, so in blue, we have African Americans, in red, we have Hispanics, and green, we have whites, right? And this is just the number of people with cognitive impairment per 1,000 people. So for every 1,000 people, usually you have twice as many people who are African American compared to whites, and 1.5 times as many Hispanics compared to whites over sort of the course of the lifespan, right? And again, that's shown here. And again, we have some ideas. We don't really understand why these sort of biological risk factors are operating the way they do. And so that's why, again, targeting these modifiable risk factors are just the key for everyone, because they're just going to help everybody until we can figure out more granularly what the actual reasons are for these more biological risk factors. And so the first one we'll talk about is blood pressure. So this is the SPRINT trial, and this was reported out fairly recently. And what they did is they had 2 strategies to manage high blood pressure. One was an intensive strategy. So the number on top, when you're talking about blood pressure, they wanted to put that one under 120. And then the standard care was to keep it under 140. And I think the new guidelines you're supposed to try and keep it under 130 over 80, I think. And so this is going down to 120, and that's actually keeping it at 140, which is kind of where it was before they moved it to 130. And they had over 9,000 people who already had high blood pressure, increased cardiovascular risk, but they did not have diabetes or dementia or a prior stroke. And what they found was that there was a significant reduction in risk of developing mild cognitive impairment or early stage type Alzheimer's or other dementias in the intensive strategy group, the 120 group compared to the standard care group. Okay, and that's really the strongest evidence to date linking treatment of high blood pressure with reduced dementia risk. So there you go. What's good for your brain is good for your heart. They're friends. Now, exercise. And again, we've heard a lot of this stuff in popular media and everything, that what this is just, data from actual studies, clinical trials, supporting these concepts, okay? This is the EXERT trial, okay? And so this is an 18-month trial. These were 300 people with mild cognitive impairment. They're, enrolled at 13 different clinics. And so they had 2 groups. One group had moderate to high aerobic exercise. And the other group had stretching, balance, and range of motion. And these were, of course, older people. I can't remember what the average age was, but it was an older population. And then the comparison group were people with mild cognitive impairment from the same clinics, but reported they just didn't exercise at all, okay? So that was sort of the control group in this case, right? And so the trial was for 18 months, but just last month they reported they did sort of a 12-month they took a peek at 12 months to see what the data was looking like. And what they found was that so far the exercise groups are showing no cognitive decline at all, whereas that MCI group that doesn't exercise at all was showing significant cognitive decline, and some even converted to dementia. So pretty interesting stuff there. And so again, exercising does a lot of things. It releases factors in your brain that promote neuron health and all sorts of things. And then one of the last 2 I want to talk about is diet, and then we'll wrap things up, or diet and cognitive resilience, and we'll wrap things up. But and so when we think about diet, we see that a lot of these studies showing that Mediterranean style diets, again, the fish, poultry, fruits and vegetables, they reduce our risk factors for AD, like high blood pressure and inflammation, and they slow cognitive decline. And so that's been shown in lots of studies. So this study kind of did the flip side of that. Okay. It was a Brazilian study. They looked at over 10,000 people and followed them over 10 years. And at the beginning of the study, they were average age 51. And they compared rates of cognitive decline in people who consumed high amounts of ultra-processed food. So that was defined as over 20% of their daily calorie intake. Okay. And so what they meant by ultra-processed foods were, foods that go through significant, processing, right, and contain large quantities of fats, sugar, salt, artificial ingredients, stabilizers, and preservatives. And the main examples listed by the participants were, the usual suspects, right? Burgers and fries, frozen pizzas, cookies, chips, sodas. And what they found at the end of the 10 years was that the high consumption group, the ultra-processed food group, eating more than 20% a day, showed a 30% faster rate of cognitive decline in memory and verbal fluency and executive function. And so you may have read about this in the press or whatever, and it was actually kind of funny. I was watching a clip of Steve Carell on his late night show, and he was telling the audience about this finding, and then he said something like, and so I only have one thing to say to the scientists: stop studying things, because you just keep taking away everything that we like every time you study something, right? It's actually kind of true in a way. But again, it just goes to show you that moderation is the key with this stuff, right? And again, the other sort of thing that helps us also understand things is, poor access to healthy foods in some of our communities, where these folks have higher risk for dementia. And this could be easily one of the reasons for that, or a contributing factor to that. Okay, and then the last thing I want to talk about is building a resilient brain. And so it turns out that activities like reading and writing letters and playing cards, the crossword puzzles, and things we've been hearing about all this time, actually do prolong brain health based on this study of over 2,000 participants. So this is from the Rush Memory and Aging Project over in Chicago. What they did is they over the course of 8 years, they had 2,000 participants, average age of 80, and they had no signs of dementia. So everybody was cognitively normal. And they found that people with the highest levels of mental activity developed dementia, if they developed it, by the age of 94, whereas people with the lowest levels of mental activity developed dementia at 86. So an 8-year difference there, just based on how much you're just, and again, a lot of this was also social interactions and social activities too, like, playing, games together and things like that. So a lot of things go into what this mental activity really means, right? But what happened was, when 695 of these people actually came to autopsy and they examined their brains, and they realized that the level of mental activity had no effect on the numbers of plaques or tangles. So in other words, even people who had high numbers of plaques and tangles, if they had a high level of mental activity, they were still living longer than people with high levels of plaques and tangles, but had low levels of mental activity. So there's something about that mental activity that was being able to sort of be resilient to the pathology that was building up in their brains. Okay. And then again, we come back. The question is, how does it work, right? So here's my little shameless plug. This is a study that I was involved in, and what they did is we took brains from 30 non-demented. So these are actually controlled, like cognitively intact elderly people. Okay, and these are postmortem brains. Half of them had low AD pathology, okay, and half of them had high AD pathology. And so we refer to those as resilient, right? So they had a lot of pathology, but they never got Alzheimer's disease or anything, okay? And then we did a technology called RNA sequencing, and that was applied to the brain samples, and it measured gene expression levels. So whether genes were high or low compared to the two groups, right? And I was telling the students earlier, Christy Kelly was the first author on this paper, and she's just really brilliant. And these are some of the graphs that she came up with based on her bioinformatics of all of this. And I don't pretend to understand it, but it just looks really cool. So I thought I would show it, right? But what we found was that the non-demented resilient subjects with the high AD pathology show greater activity of those genes that were related to the synapses, the neuron to neuron communication, and also to maintaining energy inside of the cells so that they can stay healthier and function more appropriately. Okay. And so I just want to, one last plug, as the Dementia Coalition is a key resource. And if you just can go to Michigan, midementiacoalition.org. This is a group of people from all over the state, including the UP, people at universities and organizations, aging organizations. And what we're trying to do is develop a holistic way of treating people with dementia and their caregivers and the care workers and even the local communities. And the goal is to make Michigan a dementia capable state. Okay, and there's actually a roadmap that you can download from the website, and we're working on a new roadmap right now. And there are lots and lots of resources for families who want to find, services and things related if you have a loved one who has the disease. So I just want to call attention to that. And with that, I'll stop. These are folks in the lab and collaborators at MSU and across other places. And these are the funding resources. And I'll stop there. And we only went 5 minutes over, so it's a win, right? Okay, so yeah, if anyone has any questions, and if you don't, that's great too. So.
I have one online.
Yeah, okay.
Again, if you could repeat.
Yeah.
What is the typical age that these blood tests would be administered? How would someone with a strong familial percentage get this test? We are on the second generation on siblings getting Alzheimer's at 50. Yeah, okay. So first of all, so the first question is what's the average age of people that would be getting a blood test? Well, I would say, so the average, so the majority of the largest majority of cases of Alzheimer's disease are what we call sporadic, right? They're sporadic, meaning they're just sporadic in nature. They just arise and we don't really understand why that happens. And that's like maybe 95, possibly even more % of the cases. And those tend to be after the age of 65, right? The key is, of course, after the age of 65, that's when they're getting the symptoms, right? And so if the disease is starting maybe decades before, I don't see anything wrong with getting these blood tests in middle age, right? Or even your 40s or whatever. And so the other question is, if you have a family history of people who have been diagnosed with the disease early, like in their 50s, right? So what that actually suggests to me is that there may be, so we talked about the sporadic AD being the 95%. There's the other 5% that's referred to as sort of familial Alzheimer's disease because it's genetic and runs in families. And there are a few genes that do have mutations that can cause Alzheimer's disease. If anyone ever saw the film, what was it what was that film with something about Alice? Oh, never mind. Anyway, I'll keep talking. What it suggests is that there's possibly a genetic mutation that's being inherited in the family, and you can go to I would go to a physician, a neurologist, or whatever, and talk about this. And people in the family can get tested to see if they have the mutation or not. But again, it's something that you kind of have to make to the decision. Do you want to, some people think knowledge is power, and it allows you to take care of what you need to take care of. And then, other people are just like, let's just see what happens, right? And so it's a personal decision whether or not you want to know. But if you do want to know, it's possible to go get a test just with a blood sample, and they can take the DNA from that and see if there's a mutation in one of these genes that can cause the familial forms of Alzheimer's disease. Yeah. What was that movie with? Oh God, never mind. I'll stop talking about it. Still Alice, thank you. Jeez. Okay. Yeah, and if you saw Still Alice, that's a similar situation. The
the siblings all took tests, and it turned out that some of them, some of them had the mutation, some of them didn't, and so that was kind of how that worked. But
Right now, I mean, I'm just trying to, I'm wondering about the utility of the findings from that test would be for a patient. Obviously knowing, they're not knowing and preparing one way or another, but I mean, do they currently treat them differently, like more aggressively?
Yes. Yeah, and so again, they might be perfect candidates for one of these clinical trials, right? Because it's now we're talking about prevention, right? If there's a really high, I mean, and so a person with a mutation is who doesn't have the symptoms yet would be sort of the definition possibly of preclinical AD, other than someone who just, has some markers on a brain scan. And that's the other thing, is that they could go to a specialty center like University of Michigan. It would be, if the person's close to there, or if they're not in the state, then if there's a specialty center near them, or just give the center a call if they're not anywhere close, and just explore that possibility. Yeah. Okay.
There's no other questions. I just want to say, again, get it a little closer so the mic, nope, you leave it on there.
Okay.
Just want to thank you all for coming out tonight. Again, thank you to our host, Lake Superior State University. Thank
Go
you to
Lakers.
Dr. Britt Ranson Olson, again, for the great hospitality. And I'm sure Dr. Counts will stick around a little bit in case you have some questions, but let's give him a thanks.
Oh, yeah. No, thank you. It's a pleasure. Always fun to talk about this stuff, and great awareness about it. So, yeah, thank you very much.